little bit. Buenas noches, for instance. First, I would like to thank the organizers of this meeting that they invite me to speak at your conference. Like I said, I am Diana Wittendorp, and I'm a lecturer in biology and medical laboratory science at the University of Applied Science. And that means that I teach bachelor students who will become research technicians. So now I'm going to share my screen. Hopefully it works. Okay, so my talk will mainly consist about in 3 parts. So that means I have a short introduction on bacteria and antibacterial components in human milk. The second part will be about my own research. I think the reason that I was invited to this conference is that I gave a talk for lactation, and it was translated into Spanish. And for those who are not familiar with my research, I will give a short overview of what I performed, which research I did. And since this is a conference dedicated to human milk banks, I decided to add a review on current guidelines for bacterial screening of donor human milk. So first of all, long time ago, people thought that human milk was sterile, but we found that there are many bacteria in there, and most of them they are usually there, and we call them the human milk microbiome. And there is a theory that these bacteria are actually more or less the same as the bacteria that are found in the mother's intestine, and that they are taken up by specialized immune cells, dendritic cells, and then transported to the mammary tissue. And of course, there the bacteria get into the milk and into the baby, of course, and help to establish the gut flora of the infant. So there has been also research showing that bacteria found in the mother and infant are similar. Of course, these bacteria protect the infant against pathogens and make sure that the immune system is getting mature. Other research has also shown that another role of these bacteria in the mammary tissue are not only beneficial to the infant, but also to the mother herself, in that case that it can protect against pathogens that can cause mastitis. So when we look at what kind of bacteria do we find in milk, so where do they come from? Of course, the bacteria we find are already there, so they're part of the human milk microbiome. But by handling the milk, pumping, you would also find commensal bacteria from the mother's skin and hands. And commensal means, for those of you that don't know, that they are usually living together with people, so they're supposed to be there. But of course, there could also be some other kind of contamination from the pumping equipment, or maybe the bottles or storage bags that are used, or some other kind of contamination during the pumping and handling. Freshly expressed human milk contains about 3,000 bacteria per milliliter, but there's a lot of variation. There will be milk samples that don't contain any bacteria, or they can contain even 1 million per milliliter. Most of them, which are found there, are just harmless commensals, but there can also be pathogens there, so bacteria that cause disease. And some bacterial species can be both. For example, Staphylococcus aureus is a bacterium that is present in many milk samples, and people carry the bacteria in their nose without any problem. But this bacterium is also the most common cause of mastitis. Luckily, there are also antibacterial components in human milk. For example, immune cells that can take up bacteria and kill them. And also immunoglobulins, which help activate the immune system. And there are some proteins which are also beneficial, so that's lactoferrin and lysozyme, and both of them can directly kill bacteria in human milk. But we also have to take in account that handling the milk, preparing donor milk, for example, can have an effect on these antibacterial properties of human milk. For example, if milk is frozen, the cells that are in the milk, so the macrophages and other white blood cells, they would probably be destroyed because of the formation of ice crystals. And there was also a paper in 2010 investigating that even if you leave human milk in the refrigerator, then also the white blood cell count will decrease. If you look at proteins, they are less affected by stuff you do with milk. But if you heat above 60 degrees, all proteins will be denatured, so they would lose their action. And that is what happens when you do the Holder pasteurization. And that's why some milk banks, they switch to high pressure pasteurization because it's effective in killing bacteria, but it doesn't destroy components like proteins. And also the enzymes I talked about, so the lysozyme and lactoferrin, they are only affected if you freeze and thaw the milk many times, which actually doesn't happen. But they do lose their activity if you heat it for pasteurization. So now I would talk about the research I have performed. I have to state that, for the record, I am not a researcher. I work as a teacher. But in the third year, students at our school have to do a research project lasting about 10 weeks. And for several times, I've asked students to investigate bacteria in human milk. And why did I do that? Well, I was also a volunteer for a breastfeeding organization, and I got many questions from mothers on storage guidelines. Help, I left a bottle of milk in my bag after I turned home from work on Friday. Can I still give it to my child on Monday if it's left in my bag? That kind of questions. And I looked into that and found that actually there's not so much research on this. Of course, you will probably all know the ABM protocol number 8, which is already from 2017, where the clinical guidelines for storage are stated. But if you look into that, actually these guidelines are based on a few older studies. So the research question from my students was, what is the effect of storing freshly expressed and thawed human milk in a refrigerator on bacterial count? So for those of you who are not familiar with how to measure how many bacteria there are in human milk, I made this graph. So what my students did is that they take a small sample from a bottle of milk, and then they would plate it, so spread it out over a plate with nutrient medium. Then you would incubate it overnight, and at every spot where one bacterium was there, a colony will appear. So therefore it's called colony forming units. So these colony forming units reflect the number of single bacteria in the milk. And in order to be able to count the bacteria, it might be necessary that you have to dilute the milk before in a salt solution, so saline, so you have a number of bacteria. So this is a plate from my students from last year. The OV means in Dutch undiluted. So this is the undiluted sample of milk, and this is a 10 times diluted. And so they can use these numbers and count how many bacteria there in the milk. Yes. So this is the research strategy that I used, is that we asked healthy volunteers, so usually women who work at our university or in a neighboring office, to express milk. And the students take a sample, and they would put it on a plate as soon as possible. Here it says 2 hours, but actually at the moment my students are also doing that, and sometimes they manage to do it within half an hour of expressing the milk. One part of the milk is put in the freezer for 7 days, and after 7 days it's taken out at room temperature and also put in the refrigerator, so you have a frozen thawed sample and a freshly expressed sample, which is then left in the refrigerator. And then at several time points, this plating is done to be able to count how many bacteria are there in the milk at a certain time. And we use a Malditov mass spectroscopy to identify the species which are present in the milk. Wait, I'm a bit lost. Yes. So here are the results. Here in the graph you see the time in days, and here is the bacterial count, which is in a logarithmic scale. So that means this is 1, 10, 100,000, 10,000, and 10 to the fifth. And as you probably know, that this 10 to the fifth is a kind of limit for safe consumption is being stated. So what you see is that actually during the time, the number of bacteria is not going up, it's going down. So this experiment 1 was a few years ago. I presented it a long time ago. And experiment 2 was the experiment from last year. So it's different students and different mothers. And more or less you see the same. So if you leave the milk in the refrigerator for a long time, only a few samples of mothers reach this upper limit. And some of them, this is 49 days. Some of these samples are still okay after over a month in the refrigerator. So if you have to say something about guidelines, then because there's one mother crossing the line actually somewhere between 4 and 7 days, or somewhere between 7 and 10 days, you could say that 7 days in the refrigerator is safe, which is also the current guideline. But for many other mothers, they would be able to store their milk much longer. These are the results for the frozen and thawed milk in the refrigerator. And you see more or less the same, is that the difference between the experiment 1 and experiment 2 is the timeline. Because the current guideline now says that you can only keep frozen and thawed milk for 24 hours in the refrigerator. So therefore we only studied the milk for 3 days and then we stopped the experiment. But then after we saw these results, last year we decided just to keep the milk much longer, so the thawed milk, and try to study it. And we see that here, again, this same donor of the last graph, which has already a high number of bacteria in there, her milk is safe till 4 days in the refrigerator, and then it's going above the limit. And also this donor is crossing the line somewhere between 10 and 14 days. But for some donors, you can keep even thawed milk for a long time in the refrigerator. So what I'm trying to say here is that I'm repeating this kind of research, and I'm planning to publish it, because I think maybe the current guidelines are a bit too strict. And this is for, of course, healthy mothers with healthy full-term babies. So now the last part of my talk, I would like to talk about the recommendations for bacterial screening of human milk, which probably you all know about because you work with in the Human Milk Bank. This paper is from Weaver et al. In 2019, and it has the recommendations for human milk banks from the European Milk Bank Association, EMBA. And I would like to quote a small part from that paper. And they state, so I quote, "There is a lack of published evidence to inform decision making, and the working group members concluded that to maximize safety of vulnerable immune compromised recipients, best practices suggest these guidelines." So these guidelines are not really based on published evidence. The guidelines are that all pools of milk have to be tested before pasteurization and also after pasteurization. And the screening criteria or acceptance criteria, which the EMBA advises, are there should not be more than 10 to the 5th CFU per milliliter of non-pathogenic organisms, like the normal skin flora, and there should not be any pathogen. And if it doesn't meet the criteria, it should be discarded. And after pasteurization, there should not be any growth at all. But in this paper, it's also shown in this graph that there are differences of different practices. And the EMBA says it's okay because we don't really know what is a good limit or not. So you can see that even some milk banks, so in Sweden, they say for just normal skin flora, it doesn't matter how many bacteria are in there, but they do have criteria for how many pathogens there could be in there. And just before I took this talk, I had a quick look at the posters, which are in Spanish, so I don't understand. And I saw one poster, which was also about the species which are found in human milk. And I thought that in that paper, they have this upper limit of 10 to the 5th for normal skin flora and 10 to the 4th for pathogenic bacteria. There's also the milk banks in the United States that do not test at all before pasteurization. So there is lots of variation in criteria which are being used. So even a more recent paper by Contopodi from 2021, so last year, they also tried to check what is being done in the human milk banks in Europe. So they sent a web-based questionnaire to many milk banks, and 123 milk banks replied, and then they put all this data in a graph to see what is the practices of all these milk banks. So this is a very large table. I'm not expecting you to watch everything. You can just to check the paper yourself, but there's a lot of variation in which guidelines are being used. So if I conclude this table, you can see that some milk banks are very strict. They don't accept milk which has bacteria over 100, which is quite a lot. And there's also milk banks, like I said before, if it's just normal skin flora, they don't have an upper limit. And for the pathogens, although EMBA says there should not be any at all, there are different guidelines saying that we accept a small amount of some of the pathogens like Bacillus, S. aureus, and Pseudomonas, for example. If we look at all these practices, there are some pros and cons, of course. There are also milk banks, for example, most of them in Norway, some of them in Sweden and Germany, that do not pasteurize the milk. So they give raw milk to infants. And of course, they do the bacterial screening. And that has an advantage because if you don't pasteurize, you would not lose these beneficial human milk components. But there's also a risk because pasteurization is not only to destroy bacteria, but also to destroy viruses. So there might be a risk of a viral infection. We saw that the milk banks from the United States only screen their milk after pasteurization, and only they did. That's a good thing because then you have less milk that will be discarded. But the reason why there's also pre-pasteurization screening is because there is a risk of bacterial toxins. Because bacteria that can produce toxins, those toxins are not being destroyed by pasteurization. So actually, if this is a very old paper, and it showed that in quite many milk samples there are pathogens present, and there is an average about, this is in CFU per liter, but actually this is 10 to the 4th CFU per milliliter. So that's actually the normal range of bacteria in milk. And they also state that healthy babies, they don't get sick. They can digest it without problems because of stomach acid. And I guess that's also the case. I mean, preterms also have stomach acid, although they are more immune compromised. So I did also my own research on what is normal. So these are data from the Dutch Human Milk Bank. And we also see here, so actually I copied the research before, and it also shows that 10 to the fourth is more or less common. And another thing I asked the students to investigate, is it true that for some mothers they always have a higher bacterial load and some mothers have a low bacterial load? And that's what we see here. It's a bit difficult graph. So here we see all the different donors that donated at least 5 different batches. And here is the percentages of the batches of that particular donor with the number of bacteria. So if you check donor number 17, you can see that almost half of this mother's batches don't have any bacterial growth. And the one that have a higher bacterial growth, that's only 10 to the third, so 1,000. While donor number 12, her milk, about 20% was discarded because it had too many bacteria, and her lowest number of bacteria is 10 to the fourth. So the question is actually, do children get sick or do they die because of bacteria in milk? And I tried to find the literature, and I did find some cases, but very few. This is a paper from Japan, which found that 6 infants were infected by E. coli by one of the mothers who also donated to other infants. And this is a case study, so just one mother and one baby infected by an antibiotic sensitive Staphylococcus aureus. So yes, sometimes children can get sick by bacteria in milk. I also found this paper, which is really interesting because it's quite old, it's in 1989, but what they did is they checked every milk sample that was digested by preterm infants in the first 2 weeks of their life. And of course, there were also infants which received their mother's own milk. And usually this is not tested or pasteurized at all in milk banks. And they found that about 41% of the infants were exposed to S. aureus, which is a pathogen, and even 64 of the infants got gram-negative bacilli, so for example, E. coli. But they didn't find any adverse events, so they didn't find any children becoming ill. I'm going to skip this graph because I'm already over this time, but this is just showing that there are all kinds of pathogens that they find. So I come to my conclusions. We can see that human milk contains sometimes many bacteria, and some women it's higher than the other. We can also conclude that human milk can contain pathogens in about 10 to 15% of cases. But I could only find a few cases of disease in infants by human milk bacteria. And this paper from 1989 shows that many preterms do digest pathogenic bacteria without any problems. And actually we don't know, because in most cases the mother's own milk, if a mother pumps her milk for her own preterm infants, that's usually not tested. We also know that I showed you that there's a lot of variation in bacterial screening methods or restrictions of human milk. And I think it's also the case that that strict guidelines may lead to unnecessary discarding of the human donor milk. The Dutch human donor milk has to discard about 10% of their donated milk. And we also know, of course, that if an infant is not receiving human milk, and especially preterm infants, that is also a risk for necrotizing enterocolitis, NEC. So I want to do a call out to you, the audience, that we need more research. Well, that's not that simple, because we cannot do a randomized controlled trial to see whether an infant gets ill if it gets milk with a higher or lower bacterial load. So we have to do kind of a retrospective research. So it would be really interesting to kind of repeat the research from 1989, so with the mother's own milk, and then try to find if there's a correlation with a high bacterial load and a possible illness in the infant. And if it happens, so if an infant would digest milk with pathogens, then you would see some kind of illness within 24 hours of consumption. So my take-home message for you at this conference is that I think that too strict guidelines for bacterial screening could lead to less donor milk available because you have to discard it. And if we change the guidelines to less strict, and actually some milk banks are already doing that, we need scientific research to assess how high is the risk of infection by donor milk versus the risk of not receiving donor milk. So thank you very much for your attention, and I hope I have left you with more questions than when you came to my talk. So,